Okay, I was telling Vanessa, you don't have to give me a talk every morning just to keep me at the meeting. I would stay for the whole time anyway, so 8 a.m., remember it's 5 a.m., oh actually no, 2 hours, 6 a.m. West Coast time, but anyway, no, I love this meeting, and if there's time at the end, I'll tell you a little anecdote about something really special that happened at this family meeting 4 years ago that really, to me, speaks to the importance of having conferences like this where you all get together, of course, and network and meet and support each other, but you also get to teach us, teach clinicians, teach researchers, many of whom are, studying models and flies and mice who have never seen a child or an adult with D15Q syndrome, haven't really met families who are facing some of the daily challenges or are experiencing some of the things that they're trying to understand in the lab. And I think having a platform to have all of us get together is really powerful and really rare, actually. It doesn't happen that often in other settings. And so I just want to emphasize that we learn as much, and I learn as much from all of you, not just in this setting, but in clinic every week. Probably much more than you probably learn from us. But anyway, so I know Vanessa has already, did you already talk about the clinics, or are you going to, or? You're going to, okay. So initially it said that I was talking about the clinics and trial readiness, but I really want to focus on clinical trials. I will just quickly mention that the clinic at UCLA is under an umbrella of a larger neurogenetics program that I named CARING, which is Care and Research in Neurogenetics. So we see all kids and adults who have either a known or suspected genetic cause for their neurodevelopmental condition. So kids who have developmental delay, intellectual disability, autism, other things like that, and have had genetic testing come and see us. And so in that context, we also, of course, see many, many, many families with D15Q syndrome. What's nice about the clinic is that, we do sort of an intake and a triage. We do a big diagnostic journey interview, which some of you maybe have been a part of. Maybe at next year's conference, I'll tell you a little bit about that. And then we see the families are seen by a whole team as needed. You don't need to see everyone if you don't need to see everyone, but we have genetics, psychiatry, neurology, social work, and clinical psychology. I don't know if Charlotte's here, but Charlotte does a lot of work with the families around things like augmentative communication systems, helping with IEPs, those sorts of things. And then we give sort of a comprehensive after visit summary. And the goal fundamentally, though, is to get you back to your local provider. It's not easy, and I was joking about this a couple of days ago, but it's really true. It's not easy to come to any of our clinics. It's certainly not easy to come to UCLA. And so our goal is to give you as much feedback as we can and guidance, but that then we can interface with your primary providers to help facilitate your care wherever you are. My real goal in the next year or two is to really turn our clinic and maybe some other ones into a more remote delivery model where we can do teleconferencing and provide some guidance to your local providers remotely so that we're preventing you from having to ever really fly to see us if possible. So that's something to sort of stay tuned for. But anyway, so that's our clinic. We do really specialize in the neurodevelopmental behavioral side. We do manage epilepsy for sure, but I would say that Dr. T-Bert's clinic is very much expert in epilepsy and has seen many more kids with complex epilepsies than even we have. And so sometimes we see families kind of separate out into those two clinics based on those areas of expertise. Having said that, every clinic has a neurologist who has the ability to manage, hopefully proficiently, epilepsy and behavioral and developmental issues, right? So we can help you no matter where you actually come. And just that I'd say that we have the two biggest clinics, and those are sort of the ways that we, I'd say, kind of differentiate ourselves a little bit. Okay, so I'm going to talk to you about clinical trial readiness, and I'm going to try, I know it's early in the morning and it's been a lot of lectures, and so I'm going to try to make this perhaps interactive, but at least somewhat conversational, and I'll show you some data, but the take-home points for you are that I want to really help you understand why we do clinical trials, why they're important, and why they're hard, and so why we need your help and and the help of all of us out there who are trying to run these trials to really help us move forward to get answers as to what treatments might work. So here's sort of the current treatment model for what happens in general in neurodevelopmental conditions, okay? And I use the Venn diagram below is the diagnostic criteria for autism, but don't worry about that part. It's just that in any child with a developmental condition, let's say autism as an example, currently the treatment model is we identify symptoms, right, which are behaviors. So in autism, it's social communication challenges as well as restricted interests, repetitive behaviors. We then, design a treatment, which is usually behavioral intervention, and then we look to see if those core symptoms improve. And no matter what the cause is of the set of behaviors, this is kind of the model, right? I mean, we may use medications as well, but really it's this kind of pretty crude model. The limitation with this is that there's so many different causes for having that list of symptoms, kind of how we talked about yesterday, and we'd like to move to a place where we can be a little more targeted, a little more specific, and think about, are there subgroups of kids and adults? I'm gonna say kids a lot, but recognize that I mean the whole spectrum of ages. But are there a group of kids that maybe are more amenable to a certain kind of treatment, right? And is that based on their cause? Is it based on their symptoms? And might we, with that, be a little more successful in the kinds of treatments that we're trying? And this is where genetics has been really important for us. So what we know now is that in neurodevelopmental conditions overall, 20% of kids have a known genetic cause, like DUP15Q. So when we have now a condition like DUP15Q where we know the cause, it opens the door for what I think of as really precision therapeutics. And I don't just mean gene modifying therapies, which you heard a little bit about over the last couple of days, but even precision from the standpoint of targeting symptoms that are more specific to this group of children, right? So as opposed to saying my child has autism, you can say my child has autism and DUP15Q. That means something different, right? Or at least should. And that's really the goal. And so the model, oh man, the words didn't show up on this. You're gonna have to guess what the bubbles are. I don't know why that happened. So, will they show? Okay, well, I'm just gonna tell you what these bubbles say. So now, the red box, that's funny, I've never seen this happen before. So the red box is supposed to say targeted treatments. And all the boxes, so actually I'll go through it for you. So the first box is now, as opposed to just symptoms, this is cause, right? So it says genetic oh man, sorry. This is genetic cause, which leads to changes specifically in brain function, brain development, which lead to very specific symptoms of that condition, right? So now imagine if we have a targeted treatment. The targeted treatment may not just treat the specific symptoms, but it might also be able to treat the actual underlying cause. And then the question becomes, what are our outcomes? What are we really trying to measure here that we think can change and should change with the treatment? Okay, so that's kind of that's like the dream of precision health, and I really, really believe this is the era we're in. And I'm not saying that our goal is to like eradicate every symptom, and because I absolutely believe that there's so many amazing, beautiful strengths that our kids have that we want to preserve, just that there are a lot of challenges that we'd also like to really address, right? And so if we can be more targeted in the kinds of treatments we're using and targeted in our selection of outcome measures that we want to improve, we're going to be moving in a hugely different direction. And this is where we're at, right? 10 years ago, we were mostly just at the point of figuring out what the causes were. We were just starting to do genetic testing, and we were getting lots of reports that said this variant is of unknown significance, it might be relevant, it might not be. Even with DUP15Q, we were seeing reports that said 15q duplication, maybe it's related to the developmental delay, maybe not. We're way past that now. Now we're at the point of needing treatments, right? So to have treatments, you obviously need good drug development and development of the actual therapies, but we also need to have trials that ask and test whether the treatments actually work, right? So let's take a step back and think, like, what is a clinical trial? What does it look like? So this is a simplified model of what a trial looks like, but typically in a trial what happens is you get a group of patients and they get randomized into two groups. Group number one receives the treatment, and group number two usually receives a placebo. A placebo is the treatment, a sugar pill, a fake treatment. Now, of course, if you have trials that are focused on, say, behavioral intervention, you're not going to have a placebo. That's very hard to do. So we have other kinds of designs. You can do waitlist designs where one group gets the treatment right away, another group waits and gets it a little bit later, so that way you can compare at least the kids in the waitlist portion to the kids getting the treatment right away. The reason you need these comparison groups is that you need to know if the treatment is better than whatever the child or adult might be getting anyway, right? We'd love to give the treatment up front to everyone, but then we're never going to actually figure out if the treatment really works. And so this design is really important. Of course, the goal of this is eventually to make the treatment available so that everyone can use it all the time. But you have to design the trial first and do the work to be able to figure out if the treatment's going to work. So you're split up into groups, and then the intervention is given, and at the end, you're going to have two groups with outcomes, and the question becomes, does the treatment group, or has the treatment group made more gains in the outcome of interest than the comparison group? That's what determines if a trial is successful. Does the treatment group, and I don't want to say look better because it depends what the outcome is. Is it number of seizures? Is it cognitive function? Is it, whatever the outcome might be, and we'll talk about that in a second, why that's challenging. But whatever the trial determines is the primary goal of the treatment. The question is whether that treatment group gets better. Okay, so there's a lot of challenges to this, actually. And now putting on the hat of not just someone who's involved with trials in neurodevelopmental conditions, but now think about our genetic conditions like D04C02. These trials get really hard, and we'll talk a little bit about why. So the first, So challenge number one is actually the treatment. Well, what's the treatment, right? I mean, that's something that a lot of the basic researchers are working on. We need the right treatments that might work. I would say from our standpoint, thinking about clinical trial readiness, which is what we're talking about today, we also really struggle with having measures of what we call drug target engagement, meaning measures that tell us whether the medication we're giving is actually potentially going to work. Is it hitting the target? And I'll give you an example. So a great marker of target engagement in another condition is blood sugar and diabetes, right? So if you have diabetes, your blood sugar is high, there's lots of biomarkers and objective measures we can use to determine if the diabetes is at a certain level, if it's high enough that you need treatment. When we give insulin or we make dietary modifications, we know it's working right away, well before the symptoms of maybe high blood pressure or weight or feeling really tired or those sorts of things. Before those even take effect, we know because the blood sugar goes down. And we know right away, wow, this drug is actually working, the insulin's working or another medication's working, so that blood sugar went down. We don't have measures like that in these conditions because when we're thinking about D04C02, we know it's affecting brain function, brain development, and that's a little bit harder to measure, right? But we would love to have measures of drug target engagement. And what's exciting in D04C02, as I'll show you, is we actually think we might have one with some of the EEG biomarkers that we've been identifying. But that's one of the big challenges in clinical trials in this space. The other big challenge is in selection of patients. So who are we going to actually treat? Are we going to treat the whole range of kids? Are we going to be selected in terms of which subgroup within even D04C02 we're going to treat? Are we going to treat really little young ones, or are we going to treat adults? And that becomes really challenging. Ideally, we'd love to treat as early as possible. But sometimes our treatments have safety issues. There might be concerns about whether it's going to be tolerated, especially in a very young child. So the trials are done in adults first and then scaled down to kids. But adults have very different issues and maybe very different timeframes of how they're going to make gains. And it's hard to generalize from what you learn in an adult to a little kid. So that's just the kinds of things that we're thinking about when we're designing these trials. Another big issue in trials, and I bring this up for you partly because, again, I'm sure if some of you have read about trials being done in other genetic conditions, and you'll see that many of them are deemed failures. They're, even though there's excitement about the drug, like this is true in Fragile X, this happened in many consecutive trials where there was so much excitement about the treatment, huge trials were done, and it turned out that there was no difference between the placebo and the control group. And it turned out, in Fragile X in particular, one of the biggest driving factors was that the placebo group improved. So why is that? And that's great. We want to see improvement, but if your goal is to show a difference between placebo and treatment group, and they're both making gains, your clinical trial, by definition, will be deemed a failure, right? And that's a problem if you want that drug to then get out into the community. So why might thoughts like why might a placebo group make gains? Anyone want to venture? Or I'll just answer. Yeah. Okay, great. So the point was that the drug had a sedating effect on the patient, which is really more that the treatment group maybe didn't do as well, so that the two groups are looking similar. So great thought. Two of the biggest, and that is definitely one factor, two of the biggest driving influences here are, one, that many of our outcome measures, which is the last thing we'll talk about before we get into the D04C02 specifics, many of our outcome measures rely on caregiver and clinician reports. So they're really subjective. And it doesn't mean that people are erroneously, reporting outcomes or function, but the reality is there's a lot of incentive to want your child or your patient to get better. And so we know that there is a power of belief there, and we know that just by being involved in a trial, the report of outsiders changes, and they see improvement in their, again, loved ones and their patients and those sorts of things. And so there's a big sort of biased reporting that sort of happens in both groups, but at the placebo group as well, right? So we're seeing, so that's one. And so that speaks to, again, a limitation maybe in the outcome measure. It'd be better to have more quantitative measures, objective measures that have nothing to do with someone having to report what they think. But the other piece, which I think is really important for us when we think about designing our trials, is there's a true improvement that occurs simply by being followed, simply by being paid attention to, by having experts, observing your child's development, giving feedback on that development, having, when parents fill out certain questionnaires, and maybe you guys have had this experience, they'll say to me, wow, I got asked if my child can do X, Y, and Z. It hadn't actually occurred to me to know whether my child could do that or to teach them that. So I went back and actually found out if they could do it, and lo and behold, they can do it. So next time I come in and fill out that exact same questionnaire, they can do that skill that I didn't know they could do earlier, right? So there's truly an impact of just being surveilled, if you will, and we call it the surveillance effect. And so this is important because I think it doesn't mean that the drug's not working, but it means that there's value and impact in just being involved in this kind of research. And we should be, again, as clinicians and researchers, thinking about that when we're working with our families. But again, from the standpoint of trials, it becomes, unfortunately, a challenge, right? Because our groups are converging. And in Fragile X, this is what happened. The placebo group made as much gain as the treatment group in every single clinical trial almost that was done. And so all these drugs were deemed failures, which was frustrating, actually, because we think that those are really potentially impactful. And then the last thing is outcome measures. And so with outcome measurement, the issue becomes, what are we going to measure? And this is, and I'm going to get to examples in Do15Q in a minute, but this is particularly challenging in our kids who have a range of challenges, cognitive impairment, autism, motor impairment, where, it's not that we don't see there is absolutely potential for improvement, but we're using these standardized tools that were developed frankly, for like the general population, for typically developing kids. And so our kids are not scoring in a range that is allowing us to even see meaningful change. Even though you're seeing change in specific symptoms, when we look at standardized scores, they're not budging. Right? And so I'll show you, we've been doing some work to address this, but so again, in your outcome measure, if a trial is designed for 2 months, and we're looking at something like IQ, well, IQ is probably not going to change in 2 months for any of us, unfortunately, but in general, it probably isn't, and that's what we're measuring to determine if this drug is working. That's problematic. So again, I don't mean to be so negative and to give you sort of doom and gloom that these trials are never going to work. The issue is that we're aware of this now. We've seen this happen, and in the interest of time, I didn't bring in slides to show you every trial where one of these issues has actually been a problem. But again, I keep bringing up Fragile X because it's probably the place where in genetic syndromes there's been the most trials done with mechanistic drug targets, and we've not seen success. And it's not because we don't think the drug can work, but it's frustrating, right, when the message to families is another failed trial. So we need to mitigate some of this. We need to address this, and I actually think we as a community in Do15Q can really do this. I think we can be at the forefront of it, actually, in a lot of ways. Okay, so part of our, like the overarching goal of a lot of the research we've done in Do15Q, I think of as clinical trial readiness. Am I going too slow and you're going to yell at me soon? Okay, so I'm going to keep going and you can pull me off the stage when we're done. So, but that, for me, I think that's one of the biggest take-home points is just that clinical trials are hard, but if we're aware of it, we can take some steps to address the challenges. So we've been doing a lot of work with your help over the last 4 years to better understand Do15Q. And I look at it now in the context of this is going to get us ready for trials. And there are 2 real big aspects to the research we've been doing. And I've shared some of it in meetings, but I thought it'd be good to pull it together, especially because there's a lot of new families here today. And the good news is this is all the biomarker piece, most of it is published. And the second piece, the paper is literally going to come out in the next 5 days. Like it's accepted with really minor revisions. And so we'll make sure that the print for the second, the clinical endpoints piece is given to the Alliance so you guys can have access to it. But we've been focused on 2 areas. So one is, can we identify a biomarker and Do15Q that'll help us particularly with this issue of drug target engagement. Can we have a biomarker that helps us know if drugs might be sort of hitting the target? And then the second is, let's figure out, based on doing good testing with a large group of kids, what might be the right clinical endpoints to assess when we're ready for trials. So we want to be ready as a community so that when the drugs are developed, we're there and we can have our systems in place to have a successful trial. So the research that we've been doing has been funded under a very big center grant at UCLA called the IDDRC, the Intellectual Disabilities Research Center. But we, through that, have actually been able to not just do testing at UCLA, and some of you may have come to UCLA, we've actually been able to come to the family meetings. And it's been a really incredible experience to be able to bring the research here and meet with all of you. And if some of you were in Redondo 2 years ago, I had a whole SWAT team, and we did EEG, we did gait testing, we did phenotyping, all kinds of things, and it was amazing. And we saw more families and kiddos in that 3 days than we'd seen in UCLA in a year because, you're all here, and we love bringing the research to you because it's much better for us to come to you than for you to schlep all the way to us to be involved. So that was really great. And actually, when I share this work in scientific conferences, a lot of folks have now started doing this because we've had success with it and have been going to family meetings for whatever condition is that they work with to try to bring the research to the families. Okay, so the first is the biomarker. So what is a biomarker? And this is the formal definition, but I'll just simplify it and say a biomarker is any indication of a biological process. So again, I use the diabetes example. I'm not trying to pretend that Do15Q is like diabetes or anything like that, but just think of diabetes as a great example. Diabetes is diagnosed actually largely based on the biomarker of having high blood sugar, but it's involved with all kinds of clinical symptoms, right? That are the things that we don't care about the sugar itself. We're trying to treat all the downstream implications of having really high blood sugar. But the blood sugar is our biomarker. It tells us if the treatment is working. In some cases, it might be what tells us, if there's trials where you have to have a certain level of blood glucose to be enrolled in the trial, it helps us determine who's going to be in the trial. It can be very, very helpful, okay? And so here I just list this, right? So it can help us as a measure of drug target engagement. It could possibly serve as an outcome measure. But you can imagine in neurodevelopmental conditions, biomarkers are a lot harder. And I will tell you that NIH, the government funding organization, has put millions and millions of dollars into trying to find biomarkers in autism as a whole. And I've been involved with a lot of that work. And it's really, it's not going to be actually in autism as a whole very successful because there's so much heterogeneity, so much variability, right? Which is why the work in Do15Q has been so exciting. So in Do15Q, what happened was there was a paper that came out from Larry Reiter's group earlier, it was in 2012, I think, 2013, and they were describing actually just the clinical manifestations of kids who had interstitial duplications. And so they looked at autism and cognitive function and whatnot, and in that paper, they commented on the fact that when on clinical EEG, so just the regular EEGs that all of your kids are getting for epilepsy monitoring, it was noted that there was this really interesting pattern. And again, for those of you who don't read EEGs, although you guys are probably more expert in reading EEGs than most people because you've seen so many, so I shouldn't even say that. But when you look at their clinical EEG, as a neurologist, I look at this and you can probably appreciate this kind of fast activity that you can see on the EEG. If I, as a neurologist, saw this EEG, without knowing anything about the patient, I would say this person is taking benzodiazepines. This person is taking medications that are what we call GABA, positive allosteric modulators, long-term, but basically meds that are hitting GABA or increasing levels of GABA in our brain. Like Ativan, Valium, Versed, you may have heard of those medications. If any of us took that medication, that's what our EEG would look like. So I, as someone who's been studying EEG biomarkers and autism as a whole, right, saw this and thought, this is really interesting. Because what do we know about the genetics of Do15Q? Not going to go into the details of that right now in the interest of time, but we know UB3A, this is really small, sorry, UB3A is obviously overexpressed, but there's other genes in that region that are also overexpressed, including 3 GABA receptor genes. Now, we don't know exactly what overexpression of those genes does in the brain yet, but we do have a sense that GABA, the function of GABA, which is an inhibitory neurotransmitter, GABA neurotransmission is probably altered in Do15Q. Like we have a sense of that based on what we know UB3A does and what those GABA receptor genes probably do. I'll just put it, as simply as that. So on top of that, to have this signature on EEG is really compelling because it suggests that this probably relates back to the underlying genetics of DUP15Q. So when I looked at this, I thought this might actually be a biomarker of DUP15Q. Let's investigate this. Let's not rely on, describing anecdotally what we see on clinical EEG. So that's what we did. And again, many of your kids were involved with our studies. You guys probably wore those nets and did the EEG. And so what we set out to do was quantify. We have to get some numbers on this. You have to quantify whether this signature that we see is real and is it really higher in DUP15Q compared to other kids. This signature we call beta oscillation. So beta just refers to the range of the frequency of those oscillations. So it occurs between, doesn't matter, but 15 to 30 hertz. It just reflects the speed of those brain waves, essentially. So we call it beta. And what we found was, in fact, we could quantify it, and it was really robust. And this paper came out a few years ago, and we have another paper coming out that looked at sort of the properties of this biomarker. But I'll just show you what we did in the first study was we compared kids with DUP15Q with children who were IQ, 2 minutes. I'm not going to be done in 2 minutes. Can I have 10? So what we found was this. This is really, really extraordinary, and there's no other genetic condition that has a biomarker like this, so just remember that. It's really exciting. So what we found, this is just like a heat map of the brain, and what we do, and this is just like a pretty way to show EEG data, we just looked at how much of the EEG is accounted for by those beta oscillations. And you can see red means more. This is DUP15Q. This is, so I got interrupted, but what I was saying was we compared these kids to age and IQ matched kids with non-syndromic autism. So we had a comparison group of kids who were clinically similar but didn't have DUP15Q. And then we had just age matched, typically developing kids. And so you can see kids with DUP15Q have high beta oscillations, much higher than we see in the other comparison groups. This is just another way to look at it. This is called a spectral density plot, and all you have to know is that this peak here, orange is DUP15Q. This peak of beta is clearly much higher than what we see in our other two groups. So clear biomarker of this condition. And so again, we've done lots of studies since then to think about mechanism. I'm not going to get into any of that, but I think what you guys need to know is that this actually opened a lot of people's eyes, especially in industry, meaning the pharma industry. They thought, wow, this is a biomarker. This is something that we could use when we develop drugs and DUP15Q. And let's think about how that's going to look. And oh, good, now my boxes, the title shows. So let's go back to this schematic again, right? So imagine now if we have, again, the idea of a targeted treatment, but first we have a child who has symptoms of DUP15Q when we identify that they have DUP15Q syndrome. And now we can actually do an EEG and we identify that they have these high beta oscillations. Imagine now as we start developing drugs that are targeting the altered GABA neurotransmission, which we think is important for many aspects of development, right? So you have those targeted treatments. Those beta oscillations could be a really robust, profound measure of whether the drug is hitting the target, which will open the door to us deciding whether we're going to keep going, right? In this subgroup of kids where beta changes, are those kids going to be the one we keep in the trial? It might help us determine whether this drug actually could be effective in that subgroup of kids or maybe in all of our kids. And again, this is a phenomenon that we do not have in most other conditions. This is really, really specific to DUP15Q, and I think really exciting. So this, again, has opened the door to lots of continued studies, but I wanted to just make sure you understood why we care so much about just the awake EEG. And when you guys come to UCLA and we do all the behavioral testing, why we do that 30 minutes of EEG, it's to quantify this, because we want to really, really get as much information on this as we can, again, as we get moving towards getting ready for trials. Okay, so I only have 5 minutes to talk about outcome measures, but I'm going to do it. So the other big question, though, is so what are our clinical outcomes? Because here's the thing. In the end of the day, the beta oscillations help us a lot. But we don't want to fix beta necessarily. That's an EEG. We're not treating an EEG. We're treating children. We're treating adults. We're treating symptoms. We want to improve quality of life. We want to improve overall developmental functioning and outcomes. And so we still need to figure out, if we had this kind of trial, what would be our outcome measures. And so that's really been the focus of this other real big area of work. And again, Charlotte DiStefano, who you've heard from, was really integral with a lot of this. And we've looked at a bunch of different domains. So here was this place where we really had not had large-scale clinical, as we call them, phenotyping or assessment studies in Do15Q. So we didn't even know, how do standardized measures of cognitive function, adaptive skills, how do they work in Do15Q? What do our kids look like? Where are areas of, not just areas of impairment, but how do the scores fall? Is everyone falling at the floor of certain areas so that we can't use that as a measure? Are there other measures that are giving us better signal? So that was really the question here. A lot of it was around not just understanding the clinical symptomatology, which is of course very important, but also trying to figure out which measures might be more helpful to use, again, when we develop these trials. And so I'm going to take you through this pretty quickly, because again, this paper will be out very soon. So we did measurements, and again, you guys have been probably a part of, many of you have been a part of this. We did a lot of different measures. We looked at cognition and development. We looked at adaptive skills, which is like just how does your child live in the world? What can they do? What are they challenges with? We looked at autism symptoms, and we looked at other kinds of behaviors through parent report. And I'll take you through these one by one. So cognition and adaptive skills are really a big area of focus, and I will say most clinical trials want to have some aspect of this be a part of the outcome, and it makes sense. Fundamentally, I think for many of us, adaptive skills, like how does your child live in the world, is something that we would love to see improve, and it's something that we can kind of easily measure, actually. So we looked at both. And with the cognitive ability, what we did was we used multiple measures in our kids based on their abilities, right? So we're not going to use a measure that is not appropriate for someone's developmental level. And so that means we need to mix a lot of different measures. So here's an important take-home point for you. So the way cognitive testing is done is you assess a bunch of skills, you create a score, and then you create a standard score, which is based on 100 being the quote-unquote norm, right? And that standard score is based on a population norm. So it's based on, if you tested 1,000 kids out in the community, this is where most kids would fall. That score is 100, and then you fall above and below it. These are not measures that are developed for children with Do15Q or children with global developmental delay or intellectual disability. And so what we were finding is if we turned everything into standard scores, most of our children would fall close to what we would consider sort of the floor score. That does not mean they don't have wonderful abilities and cognitive function. It means that our measures are really limited. And so what Charlotte and our team did was they modified the way they did the scoring, and they turned the scores into what we call ratio IQ scores. So the way you calculate a ratio is you take the calculated age equivalence of the child, so basically what they are scoring, based on their score, what age does that map onto? And you divide that by their chronological age, and you get a ratio. And with that, look what we were able to do. We were able to see a really nice range of scores in our kids. Had we used the standard scores, this is the floor here. We would have had no data. Like all this beautiful heterogeneity there and the range of abilities we're seeing, all these kids would have been clustered at that line. So this is really important for us because we know, again, when we move forward into doing trials, and even when we're giving you guys feedback on scores, we're going to be really thoughtful about how we measure, right, and how we calculate these. Because again, the ability is there, but it's an issue of measurement. When we looked at, and again, the group differences we won't get into. This is just the purple is interstitial and green is IDIC. And this is something that's been known for some time, that children who have interstitial duplications tend as a group to have higher scores than kids with IDIC overall. There's a lot of variability there, though. So I would say that's not, I don't want that to be the take-home point, because again, I think we do see a lot of range of scores across all our kids. The other, though, important take-home point is we also then looked at adaptive behavior, which is measured by a parent questionnaire called the Vineland, which many of you have filled out. The Vineland does really well in this with your kids. So we get a nice range of scores with the Vineland, and what we found overall, and you can see this here, right? So a nice range of scores. Don't worry about the subscores, but in all of these, communication, daily living, socialization, motor, nice range. And we found overall that adaptive skills were significantly higher than cognitive function in our kids overall. So even when there's challenges cognitively, you're teaching them really nice skills and they're picking them up. So that's important because adaptive skills might be, again, an area that it may be an area of strength, but it also seems like an area that we can leverage and maybe, again, try to even improve further with some of our treatments. So we really like this measure, and this led us to really want this to be probably one of our primary outcome measures when we do these trials. Okay, the rest, I'm going to skip that. The rest I can go over a little bit more quickly. So this just shows you what I just talked about in terms of the range of scores. So again, this is the adaptive behavior scores across this is the number of kids across the scores, and this is for IQ. And you'll see a nice distribution in the adaptive skills, whereas the IQ still tends to be shifted a little bit more to the left. We get kids clustered a little bit. So again, it just speaks to the fact that this might be a nice measure that reflects a nice range in our group of kids. With autism, the only take-home point I want to make with this slide is that every kid that we actually did an ADOS on, ADOS is the direct standardized testing for autism, met criteria. Does this mean that every child had autism, like a clinical diagnosis? No, but it means that all of our kids are showing some aspect of challenges in social communication skills. And again, I think that just means that it's a really good, important target for intervention. I won't get into this in detail. We have a paper already published on this, but what striking finding was that there were a lot of areas on the direct testing, autism testing, where kids with DUP15Q compared to those with, again, non-syndromic, so non-genetic autism, showed a lot of strengths. And it really came down to, the clinicians describe it as social motivation. We see that our kids are really socially motivated. They're interested. They want to engage. They just might not quite have the skills to do so. And their motor challenges really get in the way of that. And I'll guide you to the paper that really targeted this. But this was really important also. So autism, again, a list of symptoms based on challenges in social skills. Doesn't mean they don't want to be social. And I think having social motivation opens a big window into intervention because, it's harder to teach motivation. It's much easier to teach specific skills. And so I think, again, for all of us, when we think about the kinds of interventions we're going to be doing, we should be thinking about the fact that we can make gains in some of these social domains. But in any case, autism symptoms were definitely prevalent in this group of kids. And then I'll just say that, there's other ways to look at autism symptoms. This is a very well-known standardized scale called the social responsiveness scale that's used a lot in clinical trials. It does not work well with our kids because there's questions like this: gives unusual or illogical reasons for doing things. Well, if you have a minimally verbal child, they're not going to give you illogical reasons for doing things, and so you're going to skip that question. But the problem is when you skip a question, it affects your score. Is aware of what others are thinking or feeling. That also can sometimes be hard to discern, right, with our kids. In another checklist, which is not specific to autism but related symptoms, feels or complains that no one loves him or her, can't get his or her mind off certain thoughts. The questions like this are just not appropriate for this population overall. Again, for some kids it might be, but in a child who's minimally verbal, some of these questions are not going to be appropriate. So we know that these questionnaires, which are used a lot in autism trials, are probably not going to be useful in DUP15Q. We talked about sleep yesterday, so I will skip this, but we did get really nice data on sleep. And then I'm going to lastly say, one of the big areas that we know is a challenge in DUP15Q is motor skills. And when we went back and tried to apply some of our standardized motor scales in DUP15Q, what we found was that we were seeing, again, all our kids were falling with the same score, even though there was a lot of heterogeneity, a lot of variability. And so a colleague of mine at UCLA, who's all of her focus is on motor skills, she started asking, can we develop more quantitative, objective measures of motor function that will allow us to get some differentiation? And so what she's been looking at is the gait and gait mat, and this is, oh, you're playing it, okay, awesome. So this is not on the mat itself, but you can see how, our kiddos, there's a range, but our kiddos do struggle with gait at different, at different levels. This is just a little test where we ask them just to walk across a straight line. And this is another kiddo. with D15Q as well, again, sort of struggling with gait. And so my colleague, Dr. Wilson, thought we should quantify this. And I don't know how many of you your kids walked on the gait mat 2 years ago, but she actually has got access to gait mat technology, where it's just a mat on the ground, kids walk across it, and you can get these exquisite quantitative maps of how kids walk. So this is just a picture of just one child with D15Q and their gait, and you can see it's kind of wide base. This is like each foot is a color, right? And it's compared to a child, again, with IQ-matched autism, but not D15Q, who walks in a little bit more of a straight line, right? So in D15Q, we're seeing this difference. She looked at a huge cohort. They're writing this up right now, and I'll, again, give it to the Alliance when it's published. But, found overall that kids with D15Q do have slower walking, as well as their stride length is a little bit shorter compared to kids with IQ-matched autism. But again, just to show that we can use these technologies to develop more quantitative objective measures that are not going to be susceptible to things like placebo effect as much, right? And might really have more sensitive signal over short periods of time. So this will probably also be incorporated in any trial that we do. Promise I'm done. Okay, so now we're ending, but let's go back to this now, right? So we talked about the potential for targeted treatments, that we think about these, if we have a child with symptoms, we make the diagnosis, we use the beta oscillations as a biomarker of whether the drug's hitting the target. And then now when we think about what are the outcomes we're gonna measure? Well, we're going to measure adaptive skills. If we're going to look at cognition, we're going to use a ratio IQ score, which is much more sensitive to subtle differences between kids. We're going to look at gait. And then we didn't talk, we talked about sleep yesterday, but daytime sleepiness was one of the aspects of the sleep questionnaire where we really saw a lot of parents report issues. And it's something where we got really nice information on in all of our kids, so that may be another target. So this is where we are now, and that's exciting. Now we just need the therapies, but that's where all the basic research that's being done is really being focused. Okay, so I'll end with this. So what are our next key steps? So this is where the Alliance actually really comes in. We did use blood, sweat, and tears, as did you, to get as much information as we could from as many kids as possible, but just think there's so many more families out there that we can learn from. And the more data we have about how these different measures work, about what areas are of greatest challenge for families that we should be trying to target, what are areas of strength that we can be leveraging, the better equipped we're going to be, right? So this is really about being equipped, I think, as much as really understanding. And so that's where our big registries are important. That's where having ideally natural history studies, which some other, more, I'd say, well-developed because they've been around a lot longer. Rare disease groups have already started having natural history studies where, once a year data gets uploaded about children so we know what these conditions look like over time. Another piece that I didn't talk about today is, it's great to come to meetings and assess kids and have you guys get involved, but we really need to make this more scalable. And so we've been doing a lot of work in my group in another condition, making research and intervention delivery remote. And we use all kinds of different remote delivery. We give parents iPads, they upload information, we do Zoom conferences, we do all kinds of things in various clinical trials to make intervention and assessments more accessible. And just stay tuned for that, because we'd like to do that with D15Q as well, so that all of you can be involved with research and give us information, and we can communicate in a way that does not require all the travel. And then finally, pharma companies have started putting together working groups with families in different conditions to develop what they call these concept models, which is, what are the big areas of concern for parents? What should we be focusing on when we develop therapeutics? And I think, even though we've learned a lot from these studies, I think we also could think about having some focus groups with parents to really hone in on what are the specific areas that we'd like to focus on. Okay. So with that, I'll just, I'm done, but I'm going to say there is a trial right now, what you heard from Ovid. This is not a randomized controlled trial, so the whole placebo issue is not one. It's an open label trial for epilepsy. And I'm just going to be really clear about what the inclusion are, just in case it was not clear when they gave their presentation. It's for kids it's for IDICs, who have epilepsy and have at least 3 countable seizures a month. So if you know your child has 3 seizures a month, and there's a requirement about stable medications, but we'll just leave that to the side because that can be something you discuss with the site if you're interested in being involved. The agent itself, again, indirectly works on excitatory pathways in the brain and is thought to be potentially an effective anti-epileptic, which is well tolerated. So they want to know if it can work in D15Q. So, UCLA is a site, MGH is going to be a site soon, Minnesota's already active, and I think Georgia is already active. But if you're interested, let me know, and we can at least give you more information, because I do think the more engaged we all get in even these small trials, the more likely we are to be successful when the big trials come out, right, that are focused on some of the other symptoms that we also are really, thinking about, like the neurodevelopmental conditions. Oh, that just shows you what it looks like. So anyway, it's all ages as well. Okay, that's it. So I have to just give this 2 minute. I know Vanessa's gonna kill me. I'm like an hour over. I don't care. So 4 years ago, I wrote this piece in Spectrum News, and you guys can access it. But 4 years ago, I was at this meeting, and we had the science conference, and it occurred at the same time as the family meeting. So they didn't do them separated. And I was giving, and so in that science conference, I remember 4 years ago, it was almost all basic researchers. I don't have Guy's here because I'm gonna, oh good, I'll throw him under the bus if he's not here. So 4 years ago we had, and it was almost all postdocs, grad students, who studied flies and studied mice. They had mouse models of D15Q and Drosophila models of seizures, so they knew what flies looked like when they seized. Never seen a kid seize, but knew what flies looked like when they seized, right? So there, everyone's beautiful science is being presented, and then it was my turn to talk, and I was supposed to give like a clinical overview and then actually talk about some of the EEG works. We were just kind of getting involved with that. And I looked at the schedule, and it turned out the family parade was happening at the same time. And I looked over at Guy, who had planned, so I can blame him. He had planned the science meeting, and I said, Guy, this is ridiculous. Like, these families are here with their children and adults with D15Q, and I'm giving a lecture about D15Q syndrome. I'm not giving my talk. And we are gonna take this whole group, there's probably like 60 people, and we're gonna march on down to this family, we're gonna go to the parade, and we're gonna watch it. And so, thank you. So I wish Guy was here, because Guy was not happy about this because he had a schedule and he did not want to break the schedule. And he was like, Shefali, we have, people from all over the world here for a science meeting. And I said, I don't care. We are going to go to this meeting. And so we did. We walked down to the parade and watched it. And I will tell you, there was not a dry eye in this group of researchers, not one. And it was so moving for me, but it really was, I cannot tell you how much it impacted each of them. And I will say that when we went back to that room, our entire conversation, our entire meeting was infused with a new sense of purpose. It wasn't theoretical. It wasn't about a Drosophila having seizures that you want to stop having seizures. It was about, that child who I saw walking with their family down that runway who's been suffering from seizures for years without effective therapies, right? That's what it was about. And I just think that these are the kinds of meetings where that happens, right? And so I would say for all of you, you inspire me every day, and I'm lucky because I'm a clinician, so I get to work with you and meet with you and learn from you. But I think, we have these basic researchers who are also here who are gonna be the ones that develop these treatments since we're already with all of our other, our other pieces. So, talk to them and teach them because they want to learn, And I think the more we interface with them, the more likely it is that they're going to spend all their time trying to find the treatments, right, and trying to find ways to help all of your kids have, the best lives that they can have. So anyway, with that, I just wanted to, I'm so sorry I went so far over, but thank you for listening, and I'll end there. So.